system. So, with the anatomy, the entire process of our digestive system was in order to get nutrients out of our foodstuff. So, what we have to do in this world, in the chemical and physical digestion, is before we can actually absorb any kind of nutrients from our food, before absorption can occur, we have to break everything down. So eventually, if we're trying to get food, all of the nutrients out of our food, before any of that can really happen, you have to actually break down your what is called food stuff. We have to go from the cheeseburger to the actual mixture and then be able to pass it through our digestive tract to get our nutrients out. Essentially, what we're really talking about this is we will actually have to break down into the smallest monomer. So if you think all the way back to AMP1, when in chapter, I believe it's 3, when we did carbohydrates, when we did carbohydrates, that are our sugars. These were broken down all the way into a simple sugar. When you had your proteins were broken down all the way into an amino acid. When we looked at fats or lipids, the smallest monomer of these were going to be the fatty acids, or the fatty acid chains. And then we're also going to be looking at this with our nucleic acids. So what makes up our DNA? And these are going to get broken down into amino acids as well. It has a slightly different process than the proteins. So we're talking about breaking all these foodstuffs down into their smallest part, however we need to do that. So that's what's happening in digestion. The way this process is actually going to be helped out is with something called an enzyme. So enzymes are going to be large proteins that are going to be produced by some cell in the body, but they are considered a large protein. And what an enzyme is going to do is they will act as a biological catalyst. So a catalyst is something that was talked about in AMP1 as well. So an enzyme is going to be an example of a catalyst, sorry. So what was a catalyst? To go back and refresh our memory on what a catalyst is, a catalyst will increase the rate of the reaction. So the reaction we're looking at is going from large to small. So all of these large pieces are going to get broken down. That's a reaction. So a catalyst is going to increase the rate of that reaction. So it's going to increase the rate of the large piece getting broken down into the smaller piece. But what's cool is as it increases the rate of the reaction, it will not be used up in the process. So you will put in the catalyst, have the reaction occur, and then you will be able to have this catalyst left over. So an enzyme will be put into the reaction, and then at the end you still have the reaction. So with these are going to be called digestive enzymes. So we're going to talk about a lot of digestive enzymes today, and these are really going to be hydrolytic. So they will be involved in a water type process. So one thing that happens with an enzyme is anytime you have an enzyme, there has to be something called a substrate. So these two items are going to work together, an enzyme and a substrate. If the enzyme is a large protein that acts as a biological catalyst, then the substrate is going to be the molecule that the enzyme acts on. So essentially, if this molecule is what the enzyme is acting on, this is going to be our large foodstuff. So our large foodstuff is going to be our substrate. And then when you add water, because it's a hydrolytic digestive enzyme, then you will actually be able to add the enzyme and break everything down. You will have a very specific, this is key, you will have a very specific enzyme for each substrate. So in order to have something, an enzyme, a digestive enzyme, act on a large foodstuff, they have to fit together like a lock and a key. You have to have a very specific enzyme that will break down a very specific substrate. So we're going to talk through which specific enzymes are required for which substrates. So our substrates, let's just actually break it down like this way. We're going to start with protein. So this is the first substrate. We're going to talk about protein. So all the way back up here, when we're saying substrate, substrate, where I'm talking about this guy right here, protein. And he's going to go to this guy, an amino acid. So that is the process we're going to do first. So with a protein, when you have this, we're going to start and look at a couple things. We're going to look at the path that the protein will go down in order to be broken down. We're going to look at the enzymes that happen during this process. We're going to look at where these enzymes will actually happen in the body, because this digestive tract that we know about is quite big. So where do these enzymes work in the body? And then how do you actually absorb? What's the absorption path? So for all of our substrates, we're going to follow this. So our protein, we know that a protein starts as a, we're going to make this just a little bit more colorful to keep track of everything. So we know a protein will start as a protein, and we know that it is going all the way to an amino acid, because that's the smallest monomer of it. So what a protein actually does is it will first get broken down into a large peptide, which is peptide chain. That large peptide will get broken down into small peptides or polypeptides. If you don't know what those are, go back and review from AMP1. So our protein will go through this process of essentially getting broken down into a large piece of steak, all the way through a large peptide to small peptide or polypeptides, and then down to an amino acid, its smallest monomer. So there's a couple enzymes that will make this occur. The first enzyme will be the enzyme pepsin. Pepsin will have to work in the presence of HCl. Pepsin will only work in an acidic environment. Therefore, this is why your digestive tract will have an acidic or a low pH, is because the hydrochloric acid is needed to activate the enzyme pepsin. This enzyme will occur in the part of the digestive tract. It'll occur in the stomach. I want to note that it's what we're talking about with where we're talking about where it acts, not where it's produced. So where in the digestive tract does this enzyme act? This pepsin in the presence of hydrochloric acid will happen in the stomach. We know that to be true because our stomach is a very acidic environment. Another group of enzymes you're going to have are going to be called pancreatic enzymes. And I'm just going to do a big E for that. So pancreatic enzymes. There's a couple of these. There's going to be there's one called trypsin. There's another enzyme called chemo or chymotrypsin. And there's another one called carboxypeptidase. So this is a group of pancreatic enzymes. These guys will act in the small intestine. So notice that the name is pancreatic. That is what produces, that is where the enzyme is produced, the pancreas. Where does it actually act in the body? Small intestines. And then you have another group of enzymes that are going to be called brush border. The brush border enzymes is a family of enzymes. The brush border is in digestive intestinal tract. And a couple things you need to know from here. So the first one is going to be aminopeptidase. Aminopeptidase is a brush border enzyme. Another brush border enzyme is carboxypeptidase. Sorry. Carboxypeptidase. And then another brush border enzyme is going to be dipeptidase. And all of these enzymes are going to act in the small intestine as well. So what happens when we now have all of these enzymes breaking down our protein to our large peptide, to our small peptide, to our polypeptide, down to our amino acid? This is what we want. How do we actually absorb what we want? So our absorption path for our amino acids, we're going to have our ending monomer, our amino acid, and your amino acid will need to have sodium with it. So this is why sodium is a very important chemical for our bodies. So the amino acid and the sodium, how do we actually absorb that into our body? And the way this is going to work is these two together are going to be absorbed into the capillaries of the villi in your small intestines. So essentially, as the foodstuff travels through your small intestines, it will absorb essentially straight across the intestine wall. After you have this absorbed straight into your intestines, absorbed straight into your capillaries. Your capillaries is a blood supply. This will then be filtered by the liver. So we've taken in a new item into our blood. So then it has to go to our liver to be filtered. So one thing to look at this is the general flow we're going to do for all of our foodstuffs, is when you're looking at protein, the breakdown path, the enzymes, where it actually acts, and then the absorption of how we actually get that monomer from our digestive tract, which is a hollow tube, into our blood supply. One thing to keep in mind, you do need to know these enzymes. 100%. You need to know the enzymes, you need to know the substrate that they act upon, and you are going to need to know where in the body they occur. So a couple trends as we go, I'll show you. One trend, how do you know these enzymes relate to protein? Well, look up here. Proteins are going to be large peptides, small peptides, and polypeptides. Now look at your enzymes. Pep, 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 pep. And these two you can just hopefully remember. So when you're trying to remember what enzymes go with a protein, if there is a pep in the name, that enzyme will relate to a protein substrate. Okay, so that's proteins. We're going to do carbohydrates next. We're going to follow the exact same trend. So carbohydrates. And if you're following along with your book, I am replicating the image on page 59, which is figure 39.1. That's really all I'm doing, but I find it helpful to talk through it because otherwise it doesn't make a whole lot of sense. So we're going to start with the breakdown path. How we break down our carbohydrates. So the largest monomer of our carbohydrates is going to be a starch or a disaccharide. These are the largest, sorry y'all, the largest substrates you have. These will get broken down into a smaller one. The smaller one will get broken down and be an oligosaccharide. It could also be a disaccharide. If you're following me, the disaccharide is getting broken down to that. The starch is getting broken down to that. So just kind of keep that in mind. Then these guys are going to get broken down. They will get broken down into 3 parts. Your disaccharides will get broken down into lactose, maltose, and sucrose. So these are the 3 things that will get broken down. We're not done yet because these are not the smallest monomer. These monomers are our disaccharides. So they are made up of 2 monomers, lactose, maltose, and sucrose. Go back and review early AMP1. However, we end up with, we do have 3 monomers. So what are our monomers of the substrate carbohydrates? Here we go. Our monomers. Your maltose disaccharide will become one thing only, glucose. Maltose will become one thing only, glucose. However, lactose can also turn into glucose, and sucrose can also turn into glucose. Glucose, the monomer glucose, is going to be one of the 2 in lactose, maltose, and sucrose. So all 3 of these can get broken down into glucose. Because maltose was glucose plus glucose, so that's why it's only this one. Lactose was glucose plus galactose. So galactose plus glucose made lactose. Following me? Well, then when we have sucrose, sucrose is made up of glucose, but it's also made up of fructose. So these are our 3 smallest particles, our monomers: galactose, glucose, and fructose. Very confusing, but this is a review of AMP1. We just haven't probably revisited it in a while. So these are our carbohydrates. We're going from a starch all the way down this breakdown path. What are the enzymes that will help this process? Well, the first enzyme is going to be called salivary amylase. In salivary amylase, for all of these enzymes, we're going to talk about where they act, because that's the other part of this chart. Salivary amylase, it's in the name. Where would you expect this to act? It is going to act in the mouth. Salivary glands produce salivary amylase. And you're also going to have an enzyme that's called pancreatic amylase. Pancreatic amylase is produced in the pancreas, and it will act in the small intestines. And then you're also going to have a family of enzymes called the brush border enzymes, just like we did in protein. I'm going to do a big E for those. And there's a couple different ones you need to know. For these, you're going to have lactase is a brush border enzyme. Maltase is a brush border enzyme. Sucrase is a brush border enzyme, as well as dextrinase. And these are also going to act in the small intestine. So one thing I want to revisit real quick before we move on to the actual absorption path is these brush border enzymes. Lactase is going to be an enzyme. Enzymes have to have a specific substrate. So the enzyme lactase will act on the specific substrate of lactose. Maltase will act on the specific substrate of maltose. Sucrase will act on the specific substrate of sucrose. So this is why when people are lactose intolerant, they usually do not have the enzyme lactase because, so sorry, not maltose, lactose. Here, let's do it this way. I had the wrong one. Lactose intolerant usually do not have the enzyme lactase. So if they are lactose intolerant, they don't have the lactase enzyme to break it down. They do not break down their lactose into galactose and into glucose. Therefore, lactose builds up in their system. Their body doesn't have anything to do with it. They don't know what they can do with it. It just sits there. They're considered lactose intolerant because they can't digest it. It builds up and there are issues. So that's how that process comes together. So what is the absorption path of these enzymes? How do these work? So the absorption path. Once we have our smallest monosaccharide, so we are talking about our glucose, fructose, and galactose. Our monosaccharides will be associated with a sodium ion as well. And they will go through a process of facilitated diffusion into the capillaries. So this is important because up here in proteins, they were simply absorbed, whereas down here, there has to be facilitated diffusion. It's a slightly different process. And then these, once it's in the blood supply, they're going to be the same thing like the proteins. They're going to be filtered in the liver. So it is the exact kind of same process, but this is going to be our carbohydrates. So one trend I want to tell you about proteins was PEP. When you're looking at carbohydrates, look at LACE, salivary amylase, pancreatic amylase, lactase, maltase, sucrase, dextrinase. If there is an ACE on the carbohydrate, generally if there's an ACE, it's going to be an enzyme. That's kind of how science naming has worked. But if it has it in this context, it's a digestive enzyme. It's going to break down a carbohydrate. So that's a theme for you guys. You'll also notice that these are all produced in the small intestines. Small intestines. Look up here at proteins. Small intestines, small intestines. So that's the trend. There are two anomalies. Pepsin occurs in the stomach, and salivary amylase occurs in the mouth. So right now, the only places that digestive enzymes will work is either going to be in the stomach, the mouth, or the majority is the small intestine. So keep that in mind. Okay, moving on to the next one. We're going to do our fats. And we're going to do the same process. So our fats, how do we actually break these down? So our fats are going to be pretty easy. We're going to go from what we call, let me give this a color. We call these fat. Super easy. And they're going to get broken down into two categories. Fats will either get broken down into monoglycerides and a fatty acid. So these will go together. Or fats will get broken down into glycerol plus a fatty acid. This is the process how that works. So the enzyme, what is it that will actually make this breakdown occur? The enzyme for this is going to be bile salts. So there's nothing super exciting about bile salts, but that is going to be the enzyme. And these will occur in the small intestines. You'll notice that this one's not as complicated as all the others. Another enzyme you'll have is pancreatic lipase. Pancreatic lipase will also occur in the small intestine. So now we have our breakdown of our fat. We've got our enzymes that are going to help break it down. We know where these are going to get broken down. Now we have to know how do we actually absorb these into our body. So we have two different things, monoglycerides and then our glycerol. So they go through different things. So when you have your monoglycerides, these also are with a fatty acid, so you have to have that combination. So we're doing this one right now. Monoglycerides and fatty acids, what they're actually going to do is they will enter the intestinal wall just through straight diffusion. So nothing complicated there. They will simply just diffuse against the wall. What's different about these guys is they will end up not going straight to the liver. They will actually go to the lymphatic circulation and then get transported to the liver. So they go through a slightly different process, which is interesting. And then when we're going to have not our monoglyceride and our fatty acid, but our glycerol and our fatty acid, our second one, the process these guys are going to go through is that they will be absorbed into the capillaries. So very similar to proteins. They will be straight absorbed into capillaries, and then this will end up going to be filtered in the liver. So you'll notice that when it enters the intestinal wall, we're saying it's going into the tissue. When we're saying it's absorbed into the capillaries, it's going straight into the blood supply. So with these fats, one thing to look at is lipase. Fats are also known as lipids. How do you know which enzyme goes with a fat? Pancreatic lipase will be your helpful hint. Okay, so to wrap up this section, we're almost done. We're going to do proteins, but specifically what we're going to do when we're talking about protein is going to be nucleic acid. So how do we use the nucleic acids we need in our body? So how do we break these down? Our nucleic acids will start by being nucleic acids, and then they will get broken down into our amino acids. So this is our starting point, and these will get broken down into the smallest, which is going to be an amino acid. The enzyme that will actually make this process work is going to be a kind of a repeat of some ones that we've already talked about. You're going to have pancreatic ribonuclease. Pancreatic ribonuclease will also happen with deoxyribonuclease. These two go together, and these will also act in the small intestine. That's the where. So if we have pancreatic ribonuclease and deoxyribonuclease, what we're talking about with our nucleic acids, think DNA. We're talking about our adenine, our thymine, our cytosine, and our guanine, or in our uracil. So make sure you're thinking about those when we're talking about our nucleic acids. RNA, RN. Keep in mind, all of these are related. The other set of enzymes are going to be brush border enzymes. So this family of enzymes that specifically relate to the nucleic acids are going to be nucleosidases. Nucleosidases and phosphates. These will also break down your nucleic acids in the small intestines. Cool. So now all of these are broken down by enzymes that work in the small intestines. So that's easy to remember. What is the absorption path of these guys? So the absorption path is they will actually absorb into the capillaries. So they're going straight into the blood supply, but they're going to get absorbed via active transport. So it's not diffusion, it's not facilitated diffusion, it is active transport. Something has to carry them across. And these are going to end up also then going to the liver and getting filtered. So that's the absorption path for the nucleic acids. So what you need to know as you go through and look at exercise 39 is what are the substrates, carbohydrates, proteins, fats, or nucleic acids? What are the enzymes that are going to work on that specific substrate? So this one, this one. Where do they occur? And be able to piece together these paths. So your focus for this exercise is really just to do the same process in the same chart for a fat, for a nucleic acid, for a carbohydrate, and for a protein, to be able to do all of those.